Dear students, today talk about genetic code and protein synthesis. So contents like genetic code, protein synthesis. Actually, the protein synthesis includes two main stages. They are transcription and translation. The translation includes activation, initiation, elongation, and termination stages. We will discuss something about characteristics of genetic code. Genetic code is universal. A particular amino acid is always coded by a codon in all organisms. Example, AAA always codes for lysine in all organisms, including plants and all animals, microorganisms, etc. That means you'll consider either plants or animals, but here A always codes for lysine. It is similar in all organisms. That is under genetic code is universal. AAA cannot. That is a nitrogen basis. That is under codons. The codon is always helpful to formation of amino acids. Codons are degenerate, second character. Codons are degenerate, that means amino acids are often selected by, that's under redundancy of codons. More than one codons. Example, glycine is coded by GGU, GGC, GGA and GGG. Codons are degenerate, that means amino acids are often selected by more than one codons. Example, glycine is coded by GGU or GGC or GGA or GGG. Here only third base is different and is called Ubal base. Third one is a third base is different and it is called Ubal base. Genetic code is triplet nature. Genetic code is a triplet code. That means it needs three nitrogenous bases. So A always codes for lysine. Triplet in nature. Genetic code is commaless. Selection of amino acid takes place continuously, utilizing all bases of mRNA without a break. The number of bases on mRNA is left utilized between two codons. So, genetic code is commaless, continuous. The association of nitrogenous bases to form amino acids is a continuous process without any breaking. That is, a genetic code is commaless. So, here characteristics of genetic code is the genetic code is universal, codons are degenerate, genetic code is a triplet in nature, genetic code is commaless. And then genetic code is non-overlapping. Particular base or nucleotide on mRNA is not involved in the formation of two adjacent codons. That means same nucleotide is not involved in the selection of two amino acids in protein synthesis. So no, genetic code is a non-overlapping. That means particular base or nucleotide on mRNA is not involved in the formation of two adjacent codons. That means same nucleotide is not involved in the selection of two amino acids in protein synthesis. Non-ambiguous. So genetic code is a non-ambiguous. That means codons are sensible in the selection of amino acids without making mistakes. That means same codon will not select two different amino acids. That's one of the important characters. Genetic code is a non-ambiguous. That means codons are sensible in the selection of amino acid without making mistakes. That means same codon will not select two different amino acids. Next one now, genetic code is initiator codon. Initiator codon means protein synthesis always begins with first codon on mRNA as AUG and which codes for amino acid methionine. That means initiator codon means protein synthesis always begins with the first codon on mRNA as AUG and which codes for amino acid methionine. Next one now, terminator codon. Genetic code is a terminator codon. That means in all organisms, termination of protein synthesis is controlled by one of the three terminator codon on mRNA like UAA, UGA or UAG. That's in the terminator codon. So, genetic codes have some characters like genetic code is universal, codons are degenerate, triplet in nature, genetic code is commaless, genetic code is non-overlapping, genetic code is non-ambiguous, genetic code is initiator codon, and genetic code is terminator codon. These are the few important characteristics of the genetic code. So first one is genetic code is universal, codons are degenerate, triplet in nature, genetic code is commaless, non-overlapping, non-ambiguous, initiator codon and terminator codon. Next one now, protein synthesis. Proteins are polypeptide chains made up of amino acids. So protein means group of amino acids, association of amino acids. So amino acids are structural and functional units of proteins. So proteins nothing but amino acids. So proteins are polypeptide chains made up of amino acids. They are involved in structural and functional organization of the cells. So proteins are involved in the structural and functional organization of the cells. According to the central dogma of molecular biology, proteins are the end product of gene action. So, proteins are the end products of gene action. That means the information present in DNA is transferred to mRNA and form mRNA to protein. So, in the first stage, DNA is transferred to mRNA and from mRNA to protein. Two stages. First one, transcription. Transcription involves the synthesis of a complementary mRNA strand on DNA template strand. The process involves unwinding of DNA double helix. Actually, transcription means synthesis of mRNA strand from DNA. But DNA is going to be separated and form DNA template strand by which mRNA can synthesize. So, complementary mRNA strand on DNA template strand. The process of transcription involves unwinding of DNA double helix. Next one, now one strand serves as a template strand, antisense strand, other strand is called sense strand. So double helix stranded DNA is separated. The separated DNA molecule, one strand behaves like a antisense strand and another one behaves like a sense strand. So two strands are separated, one is a antisense strand, another one is sense strand. Next one, now synthesis of mRNA is catalyzed by an DNA dependent RNA polymerase enzyme. So by the help of DNA dependent RNA polymerase enzyme, the mRNA can synthesize. The mRNA is separated and comes out of the nucleus for translation. So after the formation of mRNA, the mRNA is separated and comes out of the nucleus for translation. Two strands of DNA rewind. So two strands of DNA rewinds. One is a DNA template strand, another one is mRNA strand. So the diagram indicates the process of transcription. So 5 prime, 3 prime, 3 prime, 5 prime. The strands are separated. First one is the non-template strand, that is sense strand. Second strand, that is a 3 prime to 5 prime strand, that is the template strand, that is the antisense strand. So during transcription, the DNA strands are separated. One strand is the template strand or sense strand. Another one is the template strand or antisense strand. So mRNA strand is going to be produced with that of a template strand or antisense strand along with the nucleotide sequences. So this indicates the entire process of synthesis of proteins, protein synthesis process. So first one now DNA undergo replication to form RNA, then by the process of translation the protein synthesis takes place. So replication, transcription, translation. Replication of DNA takes place. By the process of transcription, RNA can synthesize. By the process of translation, the synthesis of proteins takes place. These are diagrammatic representations about replication of DNA, transcription of RNA, translation forms proteins. Next one now, process of translation. The translation is the process involved in the synthesis of proteins using the coded information present in mRNA. Genetic language is converted into protein language. The process needs ribosomes, mRNA, tRNA, amino acids, ATP, GTP, enzymes, etc. Translation is completed under 4 steps. First one now, activation and selection of amino acids. Second one, initiation of peptide chains. Third one, elongation of peptide chain. Fourth one, termination of polypeptide chain. First one, activation and selection of amino acids. It requires ATP and catalyzed by an enzyme, amino acyl synthetase in presence of magnesium ions. So activation and selection of amino acids. So it requires ATP and catalyzed by an enzyme, amino acyl synthetase in the presence of magnesium ions. Second one, initiation of polypeptide chain. Here, mRNA attaches on the ribosome. mRNAs as a complementary base sequences to 3' end of tRNA or smaller subunit of ribosome. Next one now, elongation of polypeptide chain. Here, the formation of 70S ribosome and move to reaches the other end of the mRNA. That is under elongation of polypeptide chain. The termination of polypeptide chain is a chain elongation continues the end of ribosome occupied by codon UAA, UAG or UGA that signals termination of the process. So after the transcription, after the formation of mRNA strand by the DNA template, the translation process occurs. Translation process includes the synthesis process of proteins. So translation is completed under 4 steps like activation and selection of amino acids, initiation of polypeptide chain, elongation of polypeptide chain, termination of polypeptide chain. So at the end of termination, a polynucleotide chain is formed, nothing but proteins. So translation occurs between mRNA and proteins. Here the diagram shows transcription process from DNA template. mRNA strand is going to be formed. mRNA involves in the protein synthesis, that is under translation. So transcription is the process of formation of mRNA by DNA template. Translation is the process of synthesis of proteins by the help of mRNA. So all the types of RNA are involved in the protein synthesis. The types of RNA are the mRNA, rRNA and tRNA. So all are including in the process of protein synthesis. The process of protein synthesis is known as translation process. Any clarification, any doubts about characteristics of the genetic code, characters of the genetic code, and the process of protein synthesis like transcription and translation? Send me the messages. Thank you.